Hello and welcome to this 4-part educational series titled Hot Topics in COPD from Disease Phenotypes to Targeted Therapies. Each episode in the series will be dedicated to one of the following topics: the role of type 2 inflammation in COPD pathophysiology, current evidence for the use of biologic therapies in COPD, comprehensive evaluation of the disease and patient symptoms, and lastly, evolving treatment algorithms for COPD. Welcome back to this 4-part educational series titled Hot Topics in COPD from Disease Phenotypes to Targeted Therapies. I'm Dr. Surya Bhatt, and thank you for joining me again. Let's get started. Let's go through the rationale for treatment with biologics in COPD. There are now several biologics that target specific mediators of type 2 inflammation and also a little more broadly in the form of targeting alarmins. The type 2 cytokines that are predominantly involved or predominantly identified in COPD are IL-4, IL-5, IL-13, as well as the immunoglobulin IgE. These are well established in asthma but are increasingly being recognized to have an important role in COPD pathogenesis. Biologics minimize eosinophilic and more broadly type 2 airway inflammation and result in significant reduction in exacerbations and hospitalizations. There are now several specific targets for biologics. Upstream, the alarmins are being targeted in the form of anti-TSLP medications such as tezepelumab, anti-IL-33 such as itepekimab or torozakimab, and also the ST2 receptor for IL-33 in the form of astegolimab. And then more downstream, there are specific inhibitors of the IL-4 receptor, which is shared between IL-4 and IL-13 in the form of dupilumab. And then there are anti-IL-5 monoclonal antibodies such as mepolizumab and anti-IL-5 receptor blockers such as benralizumab. Several biologics have been studied in COPD. The ones that targeted T2 cytokines include mepolizumab and benralizumab. The first biologic studies in COPD involved those of mepolizumab, which is an anti-IL-5 monoclonal antibody. The METRIX and the METREOS trials looked at those with predominant eosinophilic inflammation, and the main inclusion criterion in these studies were to have at least 2 moderate exacerbations or 1 severe exacerbation, thus identifying a high exacerbation risk phenotype. And they had to have eosinophils which were high either in the form of 300 cells per microliter in the previous year or 150 at the time of randomization. When they looked at all comers, there was a 20% reduction in exacerbations only in the predominantly eosinophilic subgroup, but overall the studies were of mixed results because they included both those with high type 2 inflammation as well as non-high type 2 inflammation. The FDA did not approve this medication in 2018. Subsequently, benralizumab, which is an anti-IL-5 receptor, was studied in the Galathea and Terra Nova trials. This also did not result in any significant reduction in exacerbation frequency at all. There was also no improvement in several of the secondary outcomes that were tested, such as lung function, symptoms, and quality of life. Benralizumab did completely deplete eosinophils, but the lack of effect on the outcomes, it made us learn that perhaps eosinophils are not the be-all and end-all in COPD and perhaps are not pathogenically as important as they are in asthma. In a subsequent post-hoc analysis, the authors of these 2 trials found that patients with elevated blood eosinophil counts greater than 300, and then those with more than 3 exacerbations in the previous year, and those receiving triple therapy, if you had this combination of all 3, they were the most likely to benefit from benralizumab 100 milligram dosing with a 30% reduction in exacerbation frequency. Subsequently, there have been more studies, partially having learned from the lessons learned from the benralizumab and mepolizumab studies, and also having a drug with a broader mechanism of action in the form of dupilumab, which targets both IL-4 and IL-13. In the Boreas study, people with high exacerbation risk in the form of at least 2 moderate and 1 severe exacerbation in the previous year, and a higher blood eosinophil count threshold of at least 300 cells per microliter in the background of inhaled triple therapy were included in the trial, and they found that the annualized rate of moderate to severe exacerbations was 0.78 with dupilumab compared to 1.1 with placebo, giving a rate ratio of 0.7 that translates to a 30% reduction in exacerbation frequency. Importantly, in this trial, there were also significant improvements in lung function to the tune of about 83 mils, and improvement in quality of life with the St. George Respiratory Questionnaire score improving by 3.3 units, and there were also less severe daily symptoms. The incidence of adverse events was similar between the dupilumab and placebo groups, and these results were replicated in the NOTUS study, the findings of which were almost identical in the form of a 34% reduction in exacerbation frequency. Based on these two studies, in September 2024, dupilumab became the first biologic approved by the FDA for use in patients with COPD as an add-on maintenance therapy for inadequately controlled COPD and an eosinophilic phenotype. There are several other biologics that are currently in late-stage testing, and these are mostly looking at trying to target a broader population than those with type 2 inflammation alone, and these are mostly targeting alarmins. Itepekimab is an anti-IL-33 monoclonal antibody, which has results for a phase 2A study, and this study resulted in a 19% reduction in exacerbation frequency, which, although clinically meaningful, was not statistically significant given that this was a phase 2 study and was not powered for an exacerbation reduction. But interestingly, when the study leads looked at a subgroup of former smokers as opposed to current smokers, they found a 42% reduction in exacerbation frequency as well as an improvement in lung function in former smokers alone. Based on these results, there are two ongoing phase 3 trials, 85-1 and 85-2, predominantly targeting former smokers with COPD, trying to lower exacerbation frequency. 85-2 also is looking at current smokers in a small subset, and both these trials are expected to read out in mid to late 2025. Tozorakimab is an anti-IL-33 monoclonal antibody, which has undergone phase 2 testing, and the results of the Frontier 4 study were recently reported in a conference. And here the COPD complex event was the primary outcome, which is defined as the first occurrence of a daily symptom event or a moderate or severe exacerbation or a study dropout. And they found that there were some promising results in the form of improving complex events. And there are now phase 3 trials that are ongoing, the Oberon and Titania trials, the results of which are expected to be reported in late 2025. Astegolimab is an anti-ST2, which is an IL-33 receptor blocker, and the phase 2 trial COPD STOP didn't significantly reduce exacerbation rate. Although there was a 22% reduction, the study was underpowered and the results were not statistically significant. Aliento is a phase 2B trial, and Arnasa is a phase 3 trial, both of which are ongoing and have completed enrollment, and again should read out somewhere in mid-2025. Tezepelumab is an anti-TSLP monoclonal antibody for which the phase 2A course trial results were reported at a recent conference. Here there was a 17% reduction in exacerbation frequency, and although the primary results were not statistically significant in this not adequately powered for exacerbations phase 2 trial, they found that with increasing eosinophil counts, the exacerbation frequency impact was greater, and around 150, the exacerbation frequency started going down, and certainly in those with blood eosinophil counts greater than 300, the results were quite marked in the form of a 46% reduction in exacerbation frequency. Again, these are all preliminary results based on phase 2 trials, and there is a phase 3 trial of this anti-TSLP monoclonal antibody that is being planned. So in summary, these are really exciting times for COPD therapeutics. I think we're finally at a point where we can soon be able to have a menu of biologics to pick from, and based on endophenotyping, target the right patient with the right biologic therapy. We have learned a lot from the initial failed trials of biologics in terms of whom to target so we can identify the right patients. An eosinophil count greater than 150 certainly is promising for several of these biologics, but 300 seems to be the golden number in COPD compared to the lower threshold in asthma. And based on all these trials so far, we already have the first approved biologic available for use in COPD. And with an improved understanding of the immunopathogenic pathways in COPD, several promising biologics are now being tested in COPD. Thank you for joining me in this Hot Topics program. You can view other episodes from this series on the landing page. For additional CME opportunities, clinical resources, and links to patient education materials, please visit us at www.exchangecme.com. Lastly, please remember to take the post-test and fill out the CME evaluation to receive your credit. Thank you.